examined three different samples given to us to determine which dog is the actual guilty dog. And today they're going to be presenting their data in groups of three of the work that they've been doing over the past week. And we will hopefully have a verdict given to us by Your Honor.
Good. Okay, now you realize prosecution, it's your responsibility to prove beyond a reasonable doubt that the defendants are guilty. And you'll have an opportunity after they present their case to provide defense. You may have cross-examination, questions which you'd like to ask the members of the prosecution, witnesses, after each one testifies. Okay? Are there any procedural questions that you have before we get started?
Do you guys have any questions?
Whoever is to proceed first, come right ahead.
So, Your Honor, they have their presentation set up on as PowerPoint slides, if that's okay, Your Honor?
That's fine. Sure.
Okay, where do
I don't know if we have the capacity to do this.
because I can hook up my computer, which has,
Looks like a.
can we hook it up through a laptop?
Yeah, we'll just give this one more shot. Sure. Here we go.
Oh.
Does that look familiar to you?
Yep.
Okay. Are these different folders on the,
Yes, they are.
which one?
Mine's like 2016.
Can't get it to move.
Is this going to be able to do PowerPoint?
I don't know.
Okay.
If this doesn't work,
Oh, it's just showing up as images.
then can that thing, maybe you could go.
Yeah, that's not going to work. They've got actual PowerPoint presentations
Okay.
to do.
Okay. So I'll exit this and you can try your,
Okay. If you can tell me what, give me the cable? Do you know which?
you want to connect your laptop up to
Yes.
this?
Sorry. It's going to be a VGA cable. Or an HDMI, whichever. Is this?
Sorry?
Is this a laptop here?
No, I think that's part of the system of the video presentations.
Would it be okay if I unplug one of the cables from there?
I'm sorry?
Would it be okay if I unplug one of the cables and hooked it up?
See, I don't want to interrupt the, there's a whole mechanism for the whole state.
Oh.
Do the video presentations and everything. So, what do you think that you need to do? You need to
Connect that, and then I can connect it to my computer.
Okay, Paul? Paul, in courtroom one, they want to do a PowerPoint presentation. Can they connect that up to the video monitor? Okay. So, you think this one? Maybe this one, right here?
I think,
Can he connect that directly up to this here?
I think he can somehow. I think under the desk there, there's a, copy. Over under the desk under there might be a port where you can plug that in.
And then it'll
And
automatically
then,
feed it?
well, I think you have to put this on, like video 6 or something. Now, I'm not exactly too terribly familiar with this either.
Well, he's got a presentation on a thumb drive right here. I'm gonna pull this out,
Oh, okay,
because it shows the pictures, but it doesn't show the presentation.
okay. Now we're,
Now we're back to the beginning.
okay.
Yeah, there's no cable there.
There's not.
Yeah, so
So,
if that's the VGA video feed.
okay,
you can- That's connected to the TV, you should be able to just hook that up,
okay.
connect it straight to this one. No, that's not the right one.
That's not the right one, huh?
Is there an HDMI cable?
So,
We've got you've got an HDMI here, yep. Well, this is an HDMI female side
Right.
here.
Is there a cable that's coming from the TV out, though, an HDMI cable coming out?
No. Nope. There's an
we're
HDMI.
on the back of the TV. Why don't we pull this out a little bit?
Yep.
It'll be easier to see what we're doing.
We got an HDMI somewhere here. We have to. Stand by, stand by. Yeah. Nope, nope, nope. So this is,
I'm pretty much gonna be no help here.
you've got, let me look at it.
That's this.
You got an input right here for that?
Right, but I don't know.
I don't know anything about this. No.
Okay.
No.
We're having some technical difficulties.
Yeah.
There's just an antenna, coaxial antenna input, that's the only thing here.
Well, we're gonna have to just make do with just having, is it okay if they stand in front of, I'd like the judge actually.
Well, I guess what you're gonna do, you can show your whole presentation on this right up on the bench,
Right.
and have your witnesses and your prosecution on the board here.
Yeah.
Watching.
Yeah, that's the only thing I can get to. So I guess we're gonna have this on the bench.
I have to check with the judge on it.
Okay. So if everything goes well, we'll load up each person's file that we discussed.
put the laptop up on the bench here?
Sure.
Probably has to be positioned so the judge can see it. And all that other pieces. So you can control that through the remote?
They'll have to manually. Try to face
All rise.
Okay, please be seated. We're going to use this as a screen, is that correct?
Yes, sorry, Your Honor.
That's fine. Okay, let's proceed.
So what we'd like to do, Your Honor, is first have our first group of witnesses representing vet tested one dog. Would it be okay if they approach your bench, sir?
Surely.
Okay, group four.
Good morning, Your Honor.
Good morning. Want to tell me what happened here?
For the past few days, two of us, Jordan, Madison, and I,
Speak up so they can hear you.
have been studying dog feces DNA from our dog to figure out if we do or do not have the dog that deposited feces on the Mary Line wall. We came to a conclusion that our dog didn't deposit feces on Mary Line lawn.
Did not.
Here are some facts that will help our case. The urease for our dog, which is labeled A, was negative and a peach color. As you can see, the next slide. As you can see, the victim dog labeled B was also negative and the color was peach.
The next slide you see is glucose. Before we did our testing on it, the liquid color was red. But as you can see now, the liquid is yellow, which means A and B are both positive. And if they were negative, the liquid would be red.
The next test we did was the motility. On the left are bottles A and on the right are bottles B. As you can see, B has a red straight line and A has more fuzzy line. B is negative and A is negative also. At first we thought motility A was positive because it was fuzzy, but then we figured out that if it was positive, then yellow would have turned completely red. MSA mannitol salts ajar is things you see right now on the slide. And as you can see, all the A's are yellow and all the B's are the original color. They all were before red. The yellow Petri dishes are positive and the red Petri dishes are negative.
Citrate is the next slide you see. Both A and B are bluish greenish. That means they are both negative. If one or both letters were positive, they would be completely blue.
The last and final slide we have are the gram stains. We put them under a microscope and it showed that feces A is purple and feces B is red. Purple is gram positive and red is gram negative. So that means the feces for our dog A and the feces for the victim's dog B are not the same.
Thank you, Your Honor.
So all in all, you ran 5 tests? And 2 of them showed that the dogs were different. Is that right?
Yes, sir.
And the other 3 did not show that the dogs were different.
Yes, sir.
Okay, so what conclusion should I draw?
Yeah, the back.
Okay. So in two tests, they were different, so I guess I should conclude that not your dog. Okay. Thank you very much. Appreciate your help. Do you have any questions for these witnesses?
When you approach the bench, Your Honor, I should
Sure.
switch to the next presentation, so group 3.
Hi, Your Honor. My name is Jessica. This is my team, Kayla and Grace.
Okay, you people speak up so they can hear you.
We are the team that received the evidence on Monday, July 18th from Lieutenant Coma. We are tasked to determine whether dog number 3 was the dog that deposited the feces on Mary Lion Line. Here are our results.
When utilized on a total of 6 tests in order to determine whether our client is innocent or guilty. Here's one. The urease test is when you see if the bacteria from the sample can break down urea. As you can see, our sample is positive, so because it is bright pink, and the suspect's result was negative, so it was peach.
Okay, which is which? Can you point that out to me without, maybe you should touch the screen.
The bright pink is our dog,
That's the first one? Okay,
and the peach color is the back.
okay. Thank you.
The citrate test is done to determine whether or not a sampled species of bacterium can use citrate as a food source. In the case that a species will test positive, the agar will turn a blue color, and a negative result will stay a green color. Both dogs number 3 and number 44 turned up a negative result.
Well, what do you conclude from that?
That both of the species of bacterium can break down citrate.
So it could be the same dog,
If it was by that test alone,
just
yes.
by the test. Okay.
In order to find out whether the bacteria is motile or not, we did the motility test, which involved us studying bacteria into the motility agar with an inoculating needle. As you can see, our sample for this test is positive. That means that the bacteria is motile. Suspect's bacteria is a negative, so it is not motile.
They were different. Which tubes should I be looking at here?
This is the red fuzzy ones are our
Uh-huh.
and the ones that look like a straight line down are the
Clearly different.
The glucose test proves that bacteria from the sample can use glucose as a food source. After you add the bacteria and leave it overnight, the beaver turns yellow, meaning it eats glucose, or stays red, meaning it doesn't. Also, as you can see, ours is yellow, so it can feed off on glucose. And the suspect is also yellow.
They're all the same, are they not?
The MSA test is done to determine whether or not a sample species of bacterium is able to use mannitol as a food source and can grow in high concentrations of salt. In the case that a test is positive, the agar will turn a yellow color, and the negative will stay in orange color. Dogs number 3 and number 44 both turned up negative.
The same.
The gram stain test for a thick cell wall. In the case that a sample species of bacterium tests positive, the bacteria will be stained purple, and a negative result will yield a red stain. Both dog number 3 and number 44 yielded a gram-negative result.
In conclusion, Your Honor, urease and motility were the two tests that results came back opposite of sample B, which is the original sample from the dog that deposited the evidence on scientific results, our expert opinion as modified students, that dog number 3 is highly unlikely to be the dog that the positive evidence.
Okay. Do you have any questions for these witnesses? Well, I guess not. Okay, thank you very much.
May I approach the bench now?
Surely. Suspect 2. Surely, come right ahead. Good morning. Explain what you people did.
We have been testing suspect on number 2 to see if it was the one that was probably supposed to be going on. And we have concluded the test that it is probably guilty.
Okay, you better explain how you came to that conclusion.
Okay. The gram test is used to determine whether or not the bacteria on the waste is gram positive or gram negative. Gram negative would be red, or pink, rod shape, and rod shape. Gram positive would be purple and dot shape. Both the culprit and suspect dog culprits turned out to be gram positive. Negative,
Okay, but they were the same.
they were the same.
Thank you.
The Urease test is used to see if the bacteria from each sample can break down area and change color accordingly. Sample A and B were both negative, as we say, stayed the same color.
Okay.
We conducted a citrate test to see whether or not either culture could use citrate as a food source. If the bacteria in the culture used citrate as a food source, the top of the blue-green color, so because we spread the culture on top. As you can see here, both cultures responded negative, so it stayed the original color,
Both samples.
both samples.
Thank you.
The motility test is used to determine whether or not the bacteria is able to move through the agar. If it is negative, the bacteria would be in a straight line and it would stay where it was put. But if it's positive, it would be fuzzy and it would go the whole thing. Every single one we tested, both the suspect and culprit were all negative.
The glucose test is used to see if the bacteria use glucose as a food source. pH in the solution will change the color of the solution. Sample A and B changed color, so the results were positive. We also conducted a mannitol salts agar test, or MSA for short, to determine whether or not each culture uses mannitol as a food source or if it can grow and reproduce in high salted areas. It started originally as a red-orange color, and it would have turned a very distinctive yellow color if it had used mannitol as a food source or grew high salt. But as you can see here, it responds
responded negative once again, and they stayed the red-orange color.
So,
Okay, so this test is the PCR test, and this test is used to identify the DNA. This is the most accurate test to identify the culprit. All three of the test subject to sample were on the same ladder as the original.
what does that mean?
It means that the dog that we had and the dog that was
No,
killed.
the ladder. Can you explain the ladder to me?
Yes. This is the ladder, and this is the original, and these are three bars. And this means that they have the same DNA.
And this leads you to what conclusion? Yeah, it's a suspect dog. Well, let me ask you a question. You're the only group that dealt with this. What test is the most reliable? You've given a lot of tests here.
We feel that this is the most reliable test for testing the DNA of the dog.
Do you have any questions for these witnesses?
I guess my only question, is it possible for any of these tests to give you a wrong answer? I mean, some kind of error creep in?
Well, yes, every test. We did each test 3 times with our suspect culprit, so, and they all scored the same, so it is very unlikely for them to have, for all of them to be wrong.
Okay.
Thank you very much.
So each of the 3 tests had the same results too for all of the different tests that you did?
Yes.
And you even did the most reliable test 3 times? No.
Oh, yes.
Yes, okay.
Thank you. You can go back to your seats. What happens next?
Your Honor, I guess now that we've presented our scientific evidence, we await your
Okay.
decision.
Well, it's very interesting because no test is 100% accurate. That's the reason I asked the last scientists that were here, which test was the most accurate. Their response was the DNA test with this ladder. And it's too bad that some of you can't see that because it's pretty apparent with the test that they've done came out accurately, is the most accurate, where there's the least likelihood of an error. So therefore, I'm going to accept the results of that test as being conclusive in this matter. That being the case, it appears that suspect number 2 is the appropriate party. And I'm now going to shift to the defense. Is there a defense you would like to put onto this matter? They've met their burden of proof that suspect number 2, more likely than not, is the culprit. No defense in this matter?
Not sure how we could argue with the experts in this case.
Well, I can't, if I didn't want how to argue.
Someone set us up.
All
Someone
science,
planted it there.
there's very little in science that is 100% accurate. There's always some degree of error. And that's, this test appears to be the most accurate of all of those which were there. And I would point out to you that on all the previous tests that they have, there's a disparity in the outcome. Yes.
I was just curious about the chain of evidence and how you're sure that that's the right dog.
Yeah. That's a good question. How do we know that the evidence is from the right dog?
So, Your Honor, we have Chief Temporino who has the evidence that he's
Here's
from.
something.
Good afternoon, Your Honor.
Good afternoon.
Not sworn yet.
We're not swearing.
Okay. Your Honor, on July 15th, one of our officers was called from Mary Lyon Res Hall on the campus of Tennessee University to investigate a matter where a dog was at large, dog off leash. Dog was observed to be defecating in the area, which is in violation of the town ordinance and in violation of our school policy as well. There was no owner or handler with the dog. The dog left feces in the area. The police were called. The police collected this property. It was then collected with all the proper procedures and kept in our evidence locker in proper containers. And then from there transferred over to the Mine Flight Lab where it undertook some analysis. After the analysis was undertaken, the evidence was returned to the university storage area, where it was removed and brought to court today to be introduced as the state's evidence.
I can understand how you gathered the evidence from Mary Lyon. What about the comparative evidence that they have? Do you have any information on that?
I do not, Your Honor.
Do you know the owner of suspect number 2 here?
Owner of the dog or the feces?
Owner of the dog.
Just wanted to clarify.
The one without the tail.
We do not know the owner, Your Honor.
Oh, okay. These people are defendants here. How did they become defendants? You don't. Okay, very good. Thank you very much, Chief.
Thank you.
Any questions for the chief by either side? Yes, you have a question.
I was just curious, did someone witness the defecation to ensure that that is exactly the stuff from the same dog that you collected, or did the officers witness that, or did like a bystander?
There was a witness who called the police and described what the dog looked like, and when the police arrived, what they were told over the radio matched the description of what they found, and it was the only feces in the area, and the dog was still in the area when the police arrived.
So you identified the dog by his chain or collar?
No, we did not identify the dog.
Okay. Thank you. Excellent. Any other witnesses?
No, Your Honor.
Okay. Well, it's clear that suspect number 2 is the guilty dog in this case, and I'm making that finding. The defense has put up no defense other than a couple of very weak questions that the scientists answered very carefully. But I would point out, had they challenged the scientists, maybe they could have found some discrepancy in the testing methods or the like. It appears that they all use the same scientific methods to do this, except for the DNA test, which one group did. And it's conclusive. There's very little room for argument. So therefore, I can find that beyond a reasonable doubt, which is a very high standard, that the forensic evidence, the scientific evidence in this case is conclusive, and dog number 2 is found guilty in this matter. Also, I would point out, it was important that the chief be here, because the court has to make sure that the chain of custody, that the evidence that comes before the court has been carefully guarded and put and marked in an envelope, and every time it's transferred, whether it's to the lab or back to the police, that a record is kept of what happened to the evidence. Because evidence can get mixed up or lost, and we want to make sure that if we're going to convict somebody of a crime, which is what the case is here, that the evidence hasn't been tampered with, that it's the original evidence that was picked up. And if you'll notice, the chief has an envelope there in his hand, which is marked and on the date and time when the matter was collected. Okay, so found. Guilty dog number 2. Whoever the owner is of dog number 2 would be responsible for violating the town ordinance in this matter. Okay?
Yes, Your Honor. We've been told that the owner of that the dog's name is Shadow.
Wouldn't be me.
You sure you wouldn't ask some questions now as a defendant?
I think someone set me up.
I don't think there's any choice but to find you guilty of this matter. For an ordinance, it would be a fine that you would pay to the town. Because it's your responsibility to keep your dog under control, and it's clearly your responsibility to make sure that your dog doesn't mess other than where it's appropriate or it's cleaned up after the fact.
Yes, Your Honor.
$25 fine.
Okay.
You can pay at the clerk's office.
Will do, thank you.
Do any of the students have any questions which they'd like to ask of the court? Does anybody have any questions that they'd like to ask? No. Well, I would simply point out another thing too. Society has become, because of all of the CSI type programs and the like, has become very cognizant of forensic evidence. And things like witnesses, and I think what the chief said at the end is very, very important. They identified the dog, they saw the dog, because you need witnesses as well as forensic evidence. Forensic evidence is fine, but it's only part of the puzzle that the court has to look at. So when you see things on television, it's things like eyewitness, they're important. Forensic evidence makes them more important. We're looking for reliability of the evidence. People's memories and people's thoughts can play tricks on their mind in terms of what they identify. A brown dog, well, there are lots of brown dogs. But the forensic evidence makes it much more certain that we have the right defendant. If you watch the news, you will see that regularly now, we have people who have been in prison who are released from prison because now the forensic techniques are much more sophisticated than they were when that person perhaps was arrested and put in jail. And the court then holds a hearing to determine if the forensic evidence means we've convicted the wrong person and send the wrong person to jail. Very important what these young people have done, because it helps the legal system make sure that we haven't had a miscarriage of justice. So I appreciate the work that you've done, and I hope you learned some good things in that class, because what you presented here is pretty sophisticated stuff. Not easy to come by. And obviously, someone directed you pretty well on how to run through these numerous tests. So I appreciate it. You should appreciate it. If you want to have a future in this, you can go to school in a science curriculum, and then go to graduate school in a forensic curriculum, and become a forensic scientist, which is needed in our society. It's a good job. I would say, however, that the working conditions that you see on television are not necessarily the working conditions under which the forensic scientists work. Blue light is important, but these modern laboratories that you see on television, most of the laboratories, scientific laboratories that I've seen the forensic scientists work in, they're crowded, there's lots of machinery, they're a little bit up here disorganized because there's so much work that goes through those laboratories. It's a hard job. They don't loll around and talk to one another. They work very, very hard. So thank you very much. Guilty. Dog too.